Hi all, Dr. Clark here again, and today we're going to talk translation. So last time we talked about transcription and a tiny bit about translation. Remember, transcription is changing the DNA code into RNA, and in really just specific regions. We're interested in genes. And the reason for transcription or transcribing DNA to RNA is because ribosomes don't recognize DNA. They can't read, they can't utilize, they can't bind to the DNA code. So today we're going to talk about the second piece of transcription and translation, and that's translation. So what do you do with, or what does your body do with that messenger RNA code? Well, here's kind of a diagram of that step. So you made a messenger RNA code. It's going to bind to the small subunit of the ribosome at ribosomal RNA site. So it's not shown here, but that's going to be a binding site on the small subunit. And then you're going to have these molecules here, which are transfer RNA, going to come in, and they're going to bind up with the codons, 3 nucleotides at a time, on the messenger RNA side. So each one of these transfer RNAs is going to have an anti-codon that's going to recognize that codon. Along with that transfer RNA, you're going to have these amino acids that are attached. And so you're going to move amino acids from the transfer RNA to a chain of amino acids, which is going to be a protein, a primary protein. Okay, and the peptide bond will be formed. That's what the P site stands for. The P stands for is peptide. The A site stands for acceptor, so it accepts a new transfer. And this is showing you a prokaryotic ribosome or prokaryotic connection because there's no E site. In eukaryotes, there's an E site, which is known as the exit site. Okay? All right, so we're going to examine this in much greater detail as we progress through translation. But I just wanted to, show you the diagram and walk you through this diagram. Okay, so back to translation. First of all, translation, you've already transcribed the DNA. So you've already made a messenger RNA strand from DNA and from a specific region of the DNA, which is denoted by a promoter and a termination phase. So between the promoter region and the transcribed termination region is going to be a gene that you're going to make a copy of. Your body will, okay, to make a messenger RNA. Then that messenger RNA is going to leave the nucleus, come into the cytoplasm, or go into association with the ribosomes on the endoplasmic reticulum, and you're going to start building proteins from that messenger RNA. The way that works is you have another sequence of RNA, which we call transfer RNA. That transfer RNA is going to be associated with the codon on the messenger RNA. So transfer RNAs are going to have the anti-codon to each of the 64 codons. Okay, so you're going to have, it's not a 1-for-1, but it's pretty close. Okay, so you're going to have one transfer RNA associated with one codon. We already talked about that ribosomal RNA sequence. That's the sequence in which the messenger RNA is going to bind to on the small segment of or subunit of the ribosome. All right. When we're talking about eukaryotic translation, eukaryotic ribosomes, which are ADS ribosomes, they're going to have a EPA site. Okay, A is going to be the first site, the acceptor site, then a P site, which is the peptide site, and then an E site, which is the exit site. Okay? If we're talking about prokaryotes, which often are going to be 70S ribosomes. Okay? They're just going to have a site, an acceptor site, and a P site, a peptide site, no E site. Okay? But regardless, it's the same premise. Okay? You have transfer RNA molecules, and they get called transfer RNA molecules because they're transferring amino acids to a growing chain of amino acids or to a growing chain to make a protein. They come in, they transfer one amino acid to the next to form a peptide bond, and this chain just goes on and on and on. After the transfer RNA loses its amino acid, it will exit the ribosome and then associate with other enzymes to gain a new amino acid, the appropriate amino acid for the tRNA. And then it can move right back into the situation and add another amino acid. We'll look at this in video form in just a second. Okay? So here's kind of how that works. You have a small subunit of the ribosome, a large subunit of the ribosome. The small subunit is going to have ribosomal RNA, which is going to bind to the messenger RNA. Right, so it has a place for the messenger RNA to lay down. You have an exit site, a peptide site, and an acceptor site. Okay? And these are where the transfer RNA are going to enter into the ribosome. All right. Now, remember back when we were talking about codons, when we were talking about transcription. We said there were 64 codons. Remember that a codon is 3 nucleotides long. Okay? Well, transfer RNA molecules have the opposite of the codon. Okay? So they recognize the opposite of the codon, or they have 3 nucleotide sequence that is the opposite of the codon. So for example, the universal codon for start is AUG, adenine, uracil, guanine. The transfer RNA molecule will have an anticodon to that, which will be U, or uracil, adenine, cytosine. Okay? So AUG, its anticodon is UAC. Okay? And that corresponds to the correct transfer RNA molecule and the correct amino acid that's attached to it. In this case, the amino acid that will be attached to the start codon is methionine. All right, so like I said before, there are 64 codons, okay? And therefore there are basically 64 anticodons. There's actually less anticodons than there are codons, but that really has to do with some transfer RNA can utilize different codons with the same anticodon. Okay? And for a better explanation of this, okay, you know, you can ask me questions in class or something like that, or shoot me a line or something like that, or look at another video. It really has to do with when you look at a codon, the first 2 nucleotides determine the codon, and the last one is, on most amino acids, is interchangeable. All right, so you can change the last nucleotide on a codon, and it doesn't change the amino acid on a lot of amino acids. Not all of them, but on a lot of amino acids. That means your anticodon does not have to be exactly the same as your codon. Okay? But regardless of that, okay, 64 codons, basically 64 anticodons. Okay? When that transfer RNA loses its amino acid, like I said before, you have activating enzymes that are going to reattach the appropriate amino acid. Okay? And back to what I said before in previous lectures. DNA is not making proteins. RNA is not making proteins. Okay? They're not making amino acids. All DNA and RNA does is give you a blueprint for the order at which amino acids need to be linked together to form a certain protein. The amino acids are already in solution. The amino acids are from the result of biochemical pathways, which are linking amino groups and R groups all together to make the 20 different amino acids. That has nothing, I shouldn't say nothing, that has very little to do with your transcription and translation. Now, you do have genes that turn on those proteins and make those proteins, so those amino acids can be constructed. Okay? But that's not what we're talking about here. We're talking about using amino acids that are already in solution and lining them up and laying them down in a specific order to make a specific protein. Okay? This is translation, okay? And it's a result of transcription first, then translation. All right, so here's that transfer RNA molecule. And you can see, remember when I was talking about the difference between ribonucleic acid and deoxyribonucleic acid? I said that ribonucleic acid are single strands. Okay? They're not double-stranded. Okay? Now, that does not mean that ribonucleic acids cannot go into a double helix. Here's an example of one that does it all the time. Okay? If you look, this runs 5' to 3'. Okay? However, it goes into a double helix in many regions of the transfer RNA. Okay? We suspect that this is the result for a couple of reasons. That if the molecule is in a double helix form, it's much less likely to break apart. It's much less likely to be damaged because the double helix form is much more conserved and it's harder to break it apart. Okay. So forming a bond with itself to form kind of a structure and a carrier molecule, that's really what transfer RNA is a carrier molecule. Because it's in solution and because it's encountering other things, it probably forms a double helix for protection. Okay. Off that OH group, okay, on the 3', you're going to have the associated amino acid. The amino acid is whatever amino acid is related to whatever the anticodon is. The anticodon is related to whatever the codon is on the messenger RNA. So the messenger RNA determines the codon, which determines the anticodon, which determines the amino acid that's attached to the 3' end. Okay. Excuse me. So that messenger RNA molecule is bound to the small subunit of the ribosome through the ribosomal RNA connection, and it's going to be moving that messenger RNA, that ribosome is going to be threading that messenger RNA through the small subunit of the ribosome 3 nucleotides at a time, or what we call 1 codon at a time. Okay? As the messenger RNA is threaded through, new transfer RNA are going to bring in the anticodon and the associated amino acid into the large subunit of the ribosome. Okay? This is happening at extreme speeds, it's very quick, okay? But it's coming in and attaching, and then the amino acid is being moved from the transfer RNA to the growing peptide chain. And this just happens over and over and over until a stop codon is reached. Okay? So like I said before, that transfer RNA is going to enter the acceptor site, move to the peptide site, and then if it's a eukaryotic cell, it's going to kick out at the E site or the exit site. Okay. So again, just to reiterate, okay, transfer RNA comes into the A site, the acceptor site, shifts to the P site. When it shifts to the P site, a peptide bond is formed between the amino acids on the transfer RNA and the growing amino acid chain. You then have that transfer RNA, which no longer has an associated amino acid, moves to the E site and is released from the ribosome complex. So here's how that process works. Okay, so methionine, that's the start codon. That's the start amino acid, and it always starts kind of in that P site so something can fit in the A site to kind of get this all fired up and get going. Okay, so leucine comes in because the anticodon is read from the codon here. Leucine comes in, and then methionine and leucine bind through a peptide bond. Okay, and then a new amino acid will come into the A site, okay, and bind to leucine, and adenine binds to leucine here at the peptoid site, and then you would have another amino acid. Or at the back end here, you would have a stop codon, which would stop and disassociate the large subunit of the ribosome and the small subunit of the ribosome. Okay? And so this is going to be moving. A couple things are moving here. First of all, the messenger RNA is being threaded through the small subunit of the ribosome. At the same time, transfer RNA are going to be coming into the A site, moving to the P site, moving to the E site, and kicking out. Okay? And it's all happening very quickly. Okay? And then, like I said, once a stop codon is reached, okay, then the protein, that indicates the end of the protein. The large subunit, small subunit fall apart, the messenger RNA is released, the protein is released. Once the protein is released into solution, into the cytoplasm, that protein can go about the normal functionality of the protein. It can be folded, okay? It can be sugar stamped, so it can make its way to the Golgi apparatus, be reconfigured, folded, have a new sugar coating put on it, especially if it's going to be something like a transmembrane protein, or if it's going to be a protein that occurs as a cell marker. Okay, these things are always going to be encountering the immune system, so they need to have self-made signals on it. Lots of different situations. Okay. Once the ribosome falls apart, okay, you still have the large subunit, small subunit, and they can reassemble once a new messenger RNA comes in contact with that small subunit, and then the large subunit will come back on top, okay, and form the entire ribosome complex. Okay, so let's watch a little video about how translation works, okay, and hopefully this will make it so it's a little more clear if you're not getting it from the diagrams and from my hand movements and my voice.
The genetic information stored in DNA is transcribed into messenger RNA and then translated into protein. Translation begins when messenger RNA binds to the ribosome. The initial transfer RNA occupies the P site on the ribosome. Subsequent transfer RNAs with bound amino acids first enter the ribosome at the A site. The complementary matching of 3 nucleotides on the transfer RNA, called the anticodon, and 3 nucleotides on the messenger RNA, called the codon, ensures the correct sequence of amino acids. The messenger RNA passes along the ribosome in short spurts of 3 nucleotides at a time. As this occurs, the initial transfer RNA is moved to the E site, and its amino acid is transferred to the second amino acid at the P site. At the same time, a new codon is presented at the A site. The initiating transfer RNA, which now no longer carries an amino acid, leaves the E site, and the next transfer RNA with a complementary anticodon enters the A site. Each time a new codon sequence moves into the A site, a new transfer RNA brings in an amino acid. The old transfer RNA, paired with the previous codon, is passed to the P site and then to the E site as the amino acid it carried is transferred to the growing amino acid chain. As the ribosome proceeds down the messenger RNA, a stop codon is finally encountered. At this point, the ribosomal complex falls apart and the protein is released into the cell.
Okay, so you can see that that was a eukaryote carrying out translation. Right? And you can tell that because there was an exit site on the large ribosome. Now we're going to watch how prokaryotes go through the exact same process.
Translation is initiated by formation of an initiation complex consisting of the smaller ribosomal subunit, the first amino acid tRNA, and messenger RNA. The larger ribosomal subunit then joins the complex. Proteins called initiation factors are also involved, but are not shown. The 70S ribosome has two sites to which transfer RNA carrying amino acids can bind. One is called the peptidyl or P site, and the other is called the acceptor or A site. The initiating transfer RNA, carrying its amino acid, binds to the P site. A transfer RNA that recognizes the next codon and carries the second amino acid then moves into the A site. The amino acid carried by the transfer RNA in the P site is then joined to the amino acid carried by the transfer RNA that just entered the A site. The ribosome now advances a distance of one codon and the first transfer RNA is released. A transfer RNA carrying the next amino acid now moves into the A site and the ribosome shifts down by a distance of one codon. This is followed by the two amino acids on the transfer RNA in the P site being transferred to the third amino acid. The ribosome continues to move along the messenger RNA, and new amino acids are added to the growing polypeptide chain. Elongation of the polypeptide is terminated when the ribosome reaches a codon that does not code for an amino acid, called a stop codon. The ribosome dissociates into the smaller and larger subunits, and the messenger RNA and protein are released.
Okay, so there you can see, prokaryote, eukaryote, it doesn't really matter. The process is very, very similar. The real difference is the size of the ribosome. With prokaryotes having a 70S ribosome, okay, it's not as big as the 80S that we see in eukaryotes. It doesn't have that exit site. It just has an A and a P site. But nonetheless, the transfer RNAs are nearly identical. The start codon is the same. The stop codons are the same. And the process works the same. This is really interesting because by knowing that this is how all proteins are synthesized, right, it gives us an idea of a system that hasn't changed in probably 1 billion years. The system really has not been altered except for maybe the size and a little bit of the binding proteins and things like that. But overall, the system has not been altered in 1 billion years, which is amazing to think, in that amount of time that evolution hasn't acted much upon the system. So it means that the system works pretty well, or at least well enough that any changes would result in a faulty system. Right? All right, so with that, I'm going to come back and I'll talk about gene expression. So now you've got how DNA is copied, transcribed into messenger RNA. That messenger RNA, how is it translated into proteins? Okay? And then we're going to talk about, well, how is it that actual genes, okay, the regions of your DNA on your code, on your chromosomes, in the region that we call loci, how are those actually translated into proteins? Okay? When we come back, we'll talk about that.